I'm very honored to be able to give this webinar to all of you. I have been well, my background is in mechanical engineering and biomechanics, and from that point, I was working on. Initially, I was working on bone fracture healing, and then I opened it up towards skeletal tissue engineering in general. And I have been moving from the more typical biomechanics towards the physiology-based modeling. So there's still a large overlap, but now it's more the physiology-based and not so much the mechanics that is playing a role in what I'm doing. So what I will be doing today is give you an overview in the coming, let's say, 3/4 of an hour on the work that we've been doing in terms of computational tissue engineering with as main application the skeletal tissue engineering. So just to start off with a very small setting of the scene, what you see here is a graph that shows you the problem that tissue engineering wants to address, and that is the increasing gap between the number of people on the waiting lists for donor organs and the amount of donors that you have. You do see the brown line in the middle, which is the amount of transplantations that are executed, because obviously many donors can give more than one organ to a patient or to multiple patients. Now, typically when people think about donor organs, they think about hearts and livers and lungs, and not so much about skeletal tissues, but there is also a demand for skeletal tissues, and the bone banks that are existing cannot cope with the demands that are there. Specifically, we focus on a niche group of patients, which are patients, children with neurofibromatosis type 1, which has some skeletal consequences, as you can see on the right. These children have severe bending of their lower legs, of the tibia, that leads to atrophic non-union, so fractures that do not heal by themselves, as fractures do heal in 95% of the cases, and that is because they have this genetic defect. So what happens is that they break their leg when they're still very tiny, when they're infants, and by the time they're 4 or 5 years old, they're big enough to get surgery, and they usually get these very heavy procedures of bone transport, where there is an Elizarov cage that is put around the bone, and then a piece of bone is actually transported to fill up the gap that has arisen in the meantime. So these patients would be helped a lot if we could actually come up with a living piece of bone or intermediate tissue that can reduce the period of treatment significantly. So tissue engineering, I guess that's familiar to almost every one of you, is a field that tries to combine principles of engineering with the life sciences to make living implants. So we don't want to make implants out of plastic or metal. We want to make living implants that can actually help the organ, if it's still there, to function, or that can actually take over the functioning of the organ entirely. Now, tissue engineering was started around the year 85 as a scientific discipline, but to date the success or the impact of the discipline has been relatively limited. In Europe, we have 5 products that are approved for use in clinics, of which one is going to withdraw its product from the market because it's simply not feasible to produce these living implants at a cost that they can recuperate from the patient. So one of the major goals in tissue engineering is actually to try and translate all the biology that we find into a quantitative process that will generate a very robust and a sustainable an implant that can be produced in a sustainable way. So, and is in many production processes, in silico models are actually a part of the R&D pipeline, and so that is what we are trying to develop, the tools that can actually help to reach a sustainable tissue engineering field. Now we do this in a very integrated way, in an integrative approach, where we closely work together very closely with the biologists and the doctors that will actually be doing the experiments and treating the patients. So what you see here is an overview of Prometheus. Prometheus is a skeletal tissue engineering division of the universities of Leuven and Liège in Belgium, where we have gathered a number of different research groups groups from biomaterials, from biomechanics, from molecular biology, orthopedics, to all work around the same problem, that of trying to come up with a solution for large and non-healing fractures. So what you see here on the left is the ground substances. We work around stem cells, the growth factors, the materials, and then we move towards the mechanisms of action. So how does this work when we combine these things, when we implant them, what happens? And then later on towards the right, you see the clinical translation, where we take it towards the process engineering, the preclinical and the clinical stage. And then at the bottom of the slide, you see enabling technologies, which are imaging, modeling, the micro nanotechnologies, and data management. And so it's in this modeling, on the modeling part, that I will be presenting today. Modeling will have applications, or will interact with all of the different groups in the pipeline, so from the ground substances all the way to the clinical translation. The way I will present the work that we're doing is in 4 groups. So I've divided it into cells, carriers, culture, and clinics. It's really in vivo simulations, but I needed another C, so clinics. I will start with cells. And if we look at cells, what are the questions that one can ask? Well, you have cells. We have stem cells from different origins, from the patient, iPS-derived cells. And when we implant them, well, or when we culture them, we give them certain cues and we push them into a certain state. And then when we implant them in the body, well, they're subjected to even more cues. And what is very important also for a regulatory point of view is that we know exactly in what state our cells will be and where they are going, so that a chondrocyte does not take the path towards permanent cartilage, where we wanted it to start going down the endochondral ossification pathway and actually induce bone formation. So we need to know what is going on with our cells. And for that, we could come up with a model that actually simulates the intracellular signaling cascades of the cells. And there are different ways that you can approach these type of models, depending on exactly the question that you want to answer. So on the far right, you see phenomenological models, where you basically start with a graph showing all the interactions that you can think of, that you have found in your studies and in the literature. And on the left, you have the mechanistic modeling, where we actually work with ordinary differential equations to go in depth on the kinetics of specific pathways. Now, the application that we've taken, or the middle ground that we've taken, is the one of the additive network. The additive modeling actually is based on Boolean models, but with some additional features, as I will explain in a little bit. What you see here is the network model that we have, where we actually, well, this is a simplified representation of some of the signaling pathways that are involved in chondrogenesis of the growth plate. You see transcription factors, you see growth factors, you see other proteins there. And what we do in first instance is actually, with the Boolean model, check if we can recapitulate the different stages of the growth plate. So in a growth plate, which is kind of the motor for growth in a bone, you have different zones. You have a zone of resting chondrocytes, and then you have a zone of proliferative chondrocytes that make sure that there is a constant renewal of the pool of chondrocytes. And then there is the pre-hypertrophic and the hypertrophic zone, where chondrocytes actually are pushed towards maturation, where they start inducing a program of apoptosis and attraction of blood vessels, and will initiate endochondral ossification. So there are 2 stable states, which are the proliferative stage, that is characterized by SOX9 expression, transcription factor, and there's a hypertrophic stage, which is characterized by the expression of RUNX2 as a key transcription factor. Basically, what we've done is we've taken this network and we've said, well, all of these boxes can be either 0 or 1, or 2 in some cases. And then there's inhibitions, which are the red arrows, and there's activations, which are the black arrows. We then initiate this network with a set of genes that seem to represent the resting zone. And then we let the network actually update itself because of all the interactions, certain things will start changing. We let it update itself until it finds some kind of steady state. And then we see here in black, you see the result for the model in terms of the presence of the growth factors and the transcription factors that you find at the bottom. So for the resting zone, you see that there is a reasonable resemblance between the experimental or the biological findings and the simulation results. And what we then do is when a cell goes from the resting zone to the proliferative zone, well, there is actually the influence of the perichondrium, so that's the cells on the outside that will start changing, secreting some factors, and that will start pushing the cells from one stage to the next one. So we had changed the expression of a certain growth factor or transcription factor, and then we let the network find steady state again, and we see that also for the proliferative state we find a steady state that is reminiscent with a high SOX9 expression of the proliferative state. Then once in biology the cells start moving towards a hypertrophy, they actually escape the influence of the PTHRP, and then they will turn into hypertrophic chondrocytes where there is a high RUNX2 expression. So again, we found a stable state with the Boolean network representing the hypertrophic state. The pre-hypertrophic state did not turn out to be a stable state, and that was a very good thing because that's also not the case in biology. So that was the simple Boolean check of whether the model contains sufficient factors to simulate the chondrogenesis process. The limitations of the Boolean model are actually that you can have only, well, on or off, and there is no continuous values that are possible between 0 and 1. And another problem is that there is no difference between fast and slow processes. So fast processes typically related to post-transcriptional modifications, slow processes typically related to mRNA and transcription. And there is no way that you can make a distinction, even though that distinction is pretty relevant for certain cases in biology. So what we basically did is we made this distinction, and we had fast processes and slow processes. And for every step that we take with the slow process, we let the fast loop, so the protein network, execute one complete update and one complete search for an equilibrium before we go to the next mRNA stage. That way we can introduce somewhat more granulation in terms of time. What we then do with these networks is look at canalization. So from where are these, when I initiate all these different boxes, so all these different transcription factors and genes with a value random between 0 and 1, where are they actually going? To what stable state will this network evolve? So we did simulations where we had large numbers of random initializations between 10 and 100,000. And then we just simply check where all these simulations are going. And you see that a lot of them are ending in some kind of non-state, which is either apoptosis or pre-differentiated state. So it's a state that is not included in the model. But then there is a big part of them that are going towards RUNX2, and a few are going to the SOX9 stable state. What you can then do is look at effects of modifications of such networks. So when you knock out or when you overexpress certain factors, well, these equilibria, they change. So cells are being pushed out of one stable state into another, and that is also something that we can check. Now, this perturbation is actually interesting because it might indicate how we should treat ourselves to obtain a certain phenotype. So if we put wind, or if we overexpress wind, or we put wind on our cells, we see that we get an increase in the basin of attraction for RUNX2, which means that if you initiate the model 100,000 times, you will get more results pointing towards a RUNX2 stable state. So this might mean, if you translate this to culture processes, that by adding wind to the medium, you might induce hypertrophy of the chondrocytes. Now, what is interesting is that with these kind of screenings, you sometimes see asymmetries. For instance, if you upregulate FGFR3, you will get an increase in the RUNX2 stable state. But if you take it away, if you downregulate FGFR3, nothing really happens. So that means that FGFR3 is very important in inducing as a phenotype, but then to maintain the cell in that phenotype, the FGFR3 is not playing a role. To show you what the effect is of the added granulation that we added through using the additive approach rather than the Boolean approach, what you see here is 4 different growth factors that we actually started overexpressing in a certain dose, and the box actually represents 10,000 simulations of that growth factor. The color indicates the attractor that it is in. So if we have BMP that we add, or if we start within a SOX9 attractor and we start adding BMP2, BMP, what you see is that you will get an increase in the activity because it becomes more green, an increase in the activity of all the simulated cases. But then at some point, part of these simulations will no longer reach the SOX9 attractor, but will actually move to the non-state. In other cases, like here, the FGFi the IGF simulations, if you start increasing the dose, you will see that gradually the activity of the SOX9 is lowering until it actually has totally left the SOX9 attractor. Now, how do you validate something like this? We started from, we used microarray data on growth plates that was available in the literature. And you can see here a list of all the different microarrays that we found. And then we tried to do an inference. So we tried to infer the network from that data. Now the problem is that these are not really many samples, and that they're quite correlated. So there's not a big variation in what they were testing. So instead of trying to infer the entire network, we went for the inference of simply the transcription factor network, because that reduced the number of variables that needed to be inferred. Since we are not bioinformaticians and we don't have a preference for any of the inference methods, we used a consensus network approach where we implemented actually 8 different inference techniques. And then we made, based on a voting procedure, we went for the least bad network, for the consensus network. So the network that actually appeared in all of these inference techniques, that was the network that we went for. This will not give you the best network, but it will give you the least bad network, probably. And based on the network that we inferred, we actually saw that there were some omissions in our network that we had. So when we compared both, we saw that there were a few arrows that seemed to be missing in our network, and there were a few unknown links that we could not retrieve also in literature, and for which we are now currently performing experiments. The way you can see how good your network is by using, for instance, a receiver operator curve, where we compare on the left-hand side our golden standard model that we inferred from the literature with the inferred based on the inferred network based on the microarray data. And on the right-hand side, as a means of comparison, you see a comparison between the inferred network from the microarray data compared to a network that was derived from an online database, the string database, where it simply took all the information that it had access to related to all the different factors, so the transcription factors that were in the network, and put everything together. The reason why this is performing way less good than this one is because here you get a lot of information that might not be relevant for the case of chondrogenesis. So that's the work in terms of the cells, and that is now being explored for the use in culture media optimization. If we think about carriers, well, the questions that we have for our biomaterials are what happens to our biomaterials once we go on and plant them in vivo, how is the biology influenced, and what are the ideal properties for our biomaterials. And we do we ask ourselves these questions because obviously we want to optimize these biomaterials. So the strategy that we follow is we did a large-scale screening experiment where we took commercially available calcium phosphate polymer scaffolds. We characterized them thoroughly because the data sheets that come from the suppliers are not always very reliable. We use for that a lot of micro and nano CT to also look at structural parameters. Then we seeded them with our cells, which are periosteum-derived cells, and we implanted them, since they're human cells, on the back of nude mice. And then we extracted them to look again with the nano CT and virtual histology at the bone formation and the formation of other soft tissues. Here you see the results or the setup of one of these screening experiments where we took information at pre-implantation states, where we also at 3 days after implantation, we explanted the samples again, and we did biological measurements to look at gene expression and protein. We did the same thing for the 12-day explant, and then at 8 weeks, we evaluated the bone formation. So these weren't all the same scaffolds, but these were in the same, using the same materials, so the same groups. And what we then did was first take a data-driven approach where we asked ourselves the question, okay, at what time point do you find information in the scaffold that is actually predictive of the amount of bone that will be formed at 8 weeks? Because that is what we want to know. We want to know, is the scaffold going to induce bone formation at the relevant time, which is 8 weeks? But we don't want to wait 8 weeks every time to see what the material is doing. And so we did a partially square regression model where we looked at the data content or the information content in the data gathered at these different stages. So the materials only, pre-implantation, day 3 and day 12. And then what we saw is that actually, if you take the explant at day 3 and you look at a number of the proteins that are present, looking at beta-catenin and PKC, looking at the SMADs, that this can actually be pretty predictive for the amount of bone formation at 8 weeks post-implantation. We performed a validation experiment where we took some of the materials that we already tested as a means of control and a few others along to see, we first let the model predict the amount of bone formation, and then we implanted it and saw after 8 weeks what actually the amount of bone was that was formed. And what we saw is that the model seemed to be doing relatively well, apart for reprobone. Now, reprobone was the only material in this set that is not a calcium phosphate collagen material, but a pure calcium phosphate material. This is a very good result, in my opinion, because it, on the one hand, shows that there is some value in these data-driven models, but there is also a big limitation, and that is because it doesn't contain any mechanistic information. You really need to stay very close to the original set for which you obtained the data. If you want to go, so this is what we are now doing. We're also including the cell properties as part of the empirical modeling process, so in order to find guides towards updating our materials. Now, if you want to have more mechanistic insight in your material behavior, well, you need to use other types of models. And what you see here is a model of the calcium phosphate degradation. So you see here a cross-section through a cylindrical sample of a calcium phosphate-based material. So the white parts are calcium phosphate grains. And here you see a finite element model of that same biomaterial. We can simulate the degradation using physical principles. What you then see is that the grains are becoming smaller because they're degrading, and in the colors you see the local concentration of calcium, which at this point cannot be measured experimentally, but which is interesting to know because, well, these local concentrations are actually the concentrations that our cells that we seed on the scaffold will be exposed to. So we made this, or we calibrated these dissolution models for various materials that we're using. And then you see over 3 weeks, for instance, that the biomaterial isn't all gone, but you see that at certain points there is a degradation that we can also observe using the CT analysis that we performed on those materials. Now, once you can calculate the local calcium concentration, you can also add a model that will then, based on this local calcium concentration, tell you what is going to happen with the biology. Here you see the representation of that model, where we have the stem cells that can differentiate into osteoblasts, that can form non-mineralized matrix, that can mineralize the matrix, that can form their own growth factors, and the cells, obviously, they can proliferate, all based on the influence of the calcium that is being dissolved from the scaffold. This is a partial differential equation model that we implemented in MATLAB or in FreeFem. And what you see here, because the study is still ongoing, you see here 3 different case studies to show proof of concept, where you have a different amount of calcium dissolution from the scaffold, low, medium, and high. And you see that the bone formation response is vastly different depending on the release rate of these calcium ions. You can have bone formation either, touching on or close to the calcium phosphate grains in the center of the scaffold, so where there's open spaces, or surrounding the scaffold. And these are 3 types of bone formations, or 3 locations of bone formation that we can also find back in our implants. This is not only influenced by calcium, but also by the growth factors that we put on it. So this is something that still needs to be put into the model. The idea where we want to go with this is something like this. If you start with different cell properties, for instance, if you have here the initial amount of cells that you can see on your scaffold that is varying, you have very low cells a low amount of cells available, or a high amount of cells available. Well, what this graph tells you is how you should adapt your scaffold to that situation. If you have a low amount of cells available, which is the black line, you see that you need a scaffold that is not releasing a lot of calcium in order to have some bone formation on the vertical axis. If you have a lot of cells available, well, then basically it doesn't matter how much calcium or how fast your scaffold releases the calcium, you will get bone formation in almost all cases, but there's still an optimization that can take place. So this is how we then would like to use the model to adapt the material to the specific circumstances of the implant that we need. The next step is then culture, and for this part, we have not worked with calcium phosphate degradable materials. We've worked with scaffolds that are titanium, that are 3D printed in titanium, so that they have a fixed, well-defined geometry. What we do in these studies that focus on the bioprocessing part is we take our cells, we put them on the scaffold, and we put them in a bioreactor, which is a perfusion bioreactor, which pushes the fluid actually through the scaffold and then back again. And so what comes out, the questions that we have with these cultures, with the bioculture, are actually related to quality control. What happens here inside the bioreactor? Because obviously this is titanium, you can't just try to take a look on the inside. You can measure, you can put sensors here, and you can sample the medium, but from that you need to extract what is going on the inside of the scaffold. So what we did there is we developed a model for the growth of neo-tissue, so the cell proliferation and the extracellular matrix production by those cells together, that we call that neo-tissue. And we made a model that simulates neo-tissue formation inside these scaffolds. And we did that using a level set method, where we basically track the interface between the neo-tissue and the void space. Now, we know from biology that cells like to be in these corners, so we had a curvature-based growth velocity. So where you have here in the corners, where you have the specific values of curvature, your speed will actually increase, and then on the flat surfaces, the speed will be very low or 0. We calibrated the model using one calibration point. We had 3 different scaffold geometries, so where the pores had different geometries, and 2 different pore sizes. We used this one calibration point to actually get our speed, the speed of growth, aligned with what we know from experiments. And then we actually saw that the results of the simulations, which are the continuous lines, actually followed the results from the experiments pretty well, even with all these nonlinearity that you see here. So this curvature-based growth mechanism did seem to have also value in our situation. Now, obviously, we add fluid. We perfuse the scaffold, so there is fluid going through the scaffold, also going through the neo tissue, because this is a porous tissue, and that will also affect the growth rate of the neo tissue. So we needed to take into account the effect of the flow-induced shear stresses. And the way we simulated that was using the Brinkman equation, where we can actually have this two phases, if you like. We have, on the one hand, the Darcy equation for the porous medium part, and on the other hand, we have the Stokes, because we have a very low Reynolds numbers, the Stokes equation for the free flow part. And these two are combined in one model, let's say, which is the Brinkman equation. So for this, we could simulate the shear stress on the inside of the scaffold, in the free flow zone at the interface and in the neo tissue. Now, obviously, apart from curvature or the geometry and the shear stress, you also have oxygen that is very important. So we added an equation for the oxygen tension, where you have diffusion, advection, and consumption. And so in the end, we had a growth rate of the neo tissue, so of the interface, which is depending on curvature, on shear stress, and on oxygen. Oxygen saying that there simply needs to be oxygen for cells to survive. And for the shear stress, well, there is an optimal value where you will have optimal growth, and then when your value of shear stress becomes too high, it's harmful and there will be no growth at all. We simulated then different situations using this model for a scaffold. You have the scaffold here. This is the flow chamber of our bioreactor. So we simulated this entire bit. And what you see here is for a scaffold placed at the bottom of the bioreactor chamber. And in pink, you see the filling of the scaffold with the neo tissue. And what you can tell is when you place the scaffold at the bottom, you have relatively high shear stress of the flow here. So there will be no filling. There's no neo tissue filling. And here you have filling of the scaffold. When you place the scaffold, this is a mistake. This should be the same fluid flow. When you place it in the middle part of the bioreactor chamber, you see that you get a much more homogeneous, filling of the scaffold. And this is also what we if you look at the experimental results, and the dots are missing, but they go in the same direction. So if you have scaffolds at the bottom, we consistently saw this gap of tissue at the bottom, which we can capture with the model. And when we place the scaffold in the middle, we also get experimentally, as we get in the simulations, a much more homogeneous filling. Now you can also explore the effect of flow rates on the neo tissue growth. And again, the experimental results are not here, but they go in the same direction as the simulations. For a very low flow rate, you get relatively little filling, because there is not much stimulation, and because of the fact that oxygen is being consumed here, and so there's little oxygen left by the time the fluid actually reaches the top of the scaffold. We are now currently performing the last experiments to incorporate other variables that are frequently used in bioprocess engineering as markers, which is the glucose and the lactate. And we also tested different shapes of scaffolds. What we see here is a comparison of two different types, which is a gyroid and a D-cup. And you see the dedicated, the experiments that were set up and the simulation results, which go in the same direction. So you have a scaffold, a gyroid, that is doing relatively well, and the D-cup geometry, which is performing very poorly, both in the simulations as in the experiments. There we can also here look at cumulative lactate, so that we can actually calibrate and validate the model for that aspect as well. So if we come back to the questions that we had, what happens inside the bioreactor, the quality control, the optimization. Let's see, for the bioreactor control, Often people think that if you want to assess filling of the scaffold, that looking at the pressure drop over the scaffold, because you can have these sensors at the inlet and the outlet of the bioreactor chamber, looking at this pressure drop will tell you something about filling. But if you look at the simulation results, you see that you get a pressure drop starting around day 10 to 12, when you start noticing the pressure drop for the gyroid. If you then look at what this means in terms of neo-tissue filling, well, you're about already almost completely filled with neo-tissue before you actually get this pressure drop. So pressure drop is not a very sensitive way to assess filling of the scaffold, not unless you can actually complement this with a model like we have here. Optimization, what you see here is the filling of different types of geometry or different types of scaffolds. So we made a series of designs for scaffold geometries, and we looked at which scaffold geometries were actually performing well in terms of neo-tissue filling or poorly in terms of neo-tissue filling. Now, when we want to do an optimization, a proper optimization, because what we did here is we just simply designed 6 scaffolds or so, and we ran the simulations. But if you want to do a proper optimization, also in terms of how frequently should I change my medium, how much of the medium should I change every time I change it, well, you need to do Bayesian optimization, for instance. And with a model like the mechanistic model that takes a few days to run, this becomes very intractable. So what we did is we made a reduced model, which essentially is a 0D version of the mechanistic model. It can still capture all the essential elements. Only what it cannot do is give you a value, give you at every location in the scaffold the value of oxygen and glucose. It will give you one general value which corresponds to what you would also measure in the medium in the bioreactor itself. And with this reduced version, which takes 3 seconds to run, you can actually do very rigorous optimization in terms of medium refreshment time, medium refreshment amount, and you can look for the optimal settings. This is one where we only looked at medium refreshment, but you can also add cost. Obviously, the more frequently you refresh your medium, well, the higher the cost. So if you take cost into account, then probably the optimal solution will shift somewhere towards this regime. But then now you have something that you can actually play with. And once we have an optimal regime with the reduced model, we can check it in the full model and we can perform the experiment. What happens in the bioreactor? Well, to answer that question, with the model, you can actually, at every location in the bioreactor, give a value for glucose, lactate, oxygen, whatever variable it is that you have in the model. And so it allows you to identify niches. It also allows you to say, well, okay, if 20% of the cells that are inside my scaffold experience a certain shift or a certain oxygen low threshold, let's say, then we need to stop the simulate, or then we need to stop the culture, or we know that some differentiation will take place, and that is not something we're interested in. So all these specific things can be taken into account when optimizing the scaffold. Now, how can we validate this or connect this again to things that we can measure? We're performing metabolomics on the medium of the scaffold so that we can look for additional markers that might be interesting. Maybe oxygen and glucose, although they're routinely used, are not the best indicators of what is going on inside the scaffold. So we are performing metabolomics to get a better idea on that. And then we're also developing the nano-CT imaging so that we can online also image what is going on inside the scaffold, even though it's a titanium scaffold. But using contrast-enhanced contrast agents, we can actually also simulate the soft tissues during culture without the need to remove the scaffold from the bioreactor. So if we then go towards the final step, so once we have a scaffold, we will implant it. When we implant it, there are questions, of course. The questions are then, what is the cause of adverse fracture healing, and how can you design treatment strategies for that? well, the model that we use for that dates back to my PhD, so a very long time ago, where we have a multiscale model which contains the relevant cells, so osteoblasts, chondrocytes, fibroblasts, stem cells, the tissues that they can make, the growth factors that they produce and that they are influenced by. We then also added the simulation of individual blood vessels, where there is oxygen secretion when the blood vessel closes a loop. And this blood vessel migration is dependent on intracellular variables, so the delta 4 notch signaling loop. Here's a representation in terms of time and space. You see that this is a multiscale model where we have the tissue level part on the one hand, and then the discrete representation of the blood vessels on the other hand. Now we applied this model, this fracture healing model, in its different variants that we have to different problems related to adverse healing. First one being the periosteal and endosteal injury, so when you have fractures with a lot of soft tissue damage. The periosteum and the marrow are affected as well, and we know that there is very poor healing. What we then try to do is see if you add stem cells at a certain point after healing. In this case, post-fracture week 3, when normally you should see already some healing occurring. If you add stem cells at that point, you get a full fracture healing at 16 weeks post-fracture. This is in rats. Now, the problem was that in the pilot experiments that we were performing, we did not see this healing, this complete healing occurring. What we saw was unicortical healing, so always on one side, but not always on the same side. So we used the model and we tested a number of different cases. So maybe if we injected it not in the center, but more towards the side or even on the outside instead of inside the callus, then you see that indeed you get this unicortical healing that we observed in the experiments. A second case of adverse healing was for the critical size defect. So when you have a normal fracture healing in a rat, it will heal in a number of weeks. You get first the direct bone formation, and then in the center you get first cartilage formation, then followed by bone formation. But when you have a large defect, you see that bone formation, it advances a little bit in the beginning and then stops at a certain point. This actually corresponds to what we see in the experiments. Even in mice that heal notoriously well, you see that after 8 weeks with this large gap, you get no bone formation. You get capping, so a bit of bone forming at the ends, as we see here, although we get too much, and then nothing in the center. Now, why is this? Well, this is because the blood vessels aren't coming in fast enough. So by the time blood vessels actually are able to provide a sufficient level of oxygen in the entire scaffold or in the entire callus, the cells that were there have died and can so no longer respond to the oxygen that is there. So we are working on designing new materials that could actually release oxygen. So they have oxygen glucose releasing microparticles, so that we maintain a certain level of oxygen through this first phase, so that the cells that are present in the center are not dying. And we used the model to calculate or to predict the minimum oxygen tension that would be required to keep a minimum amount of cells alive. And we also used the model to calculate how long this minimum level should be maintained before the biology could actually take over. And so currently the oxygen releasing beads, they have been created and they are being tested for their release profiles. And then once that is done, we can implant them. A last case of adverse healing is the mechanical case. So where we know that with normal load, we get good fracture healing, and good fracture healing, you see here the fibrous tissue going down over time, cartilage being formed and degraded, and bone being formed over time inside the fracture. Now, when we have an overloading case, you see that there is some bone formation. This is intramembranous ossification, but then the endochondral ossification is not taking place. So there is the cartilage and the fibrous tissue component are sticking around and they're not going away. Now, if we in the simulation then either add a growth factor or we stabilize the fracture. We see that in this case, for a high concentration of the growth factor, we get administered at this point, we get healing. So you see that cartilage is going down and bone endochondral ossification is starting up. When we don't add enough growth factors, even though we stabilize, what we see here is that we get also bone formation, but to a smaller extent and more slowly. So we have a fracture that probably is healed in the end, but still contains a fraction of soft tissue. The last example that I would like to talk to you about is that of the congenital disordered NF1 that I started the presentation off with. So we also applied this bone regeneration model to this NF1 case, where we know from the literature and from the experts that this NF1 mutation affects a certain number of biological processes that are relevant for bone regeneration. And I've listed all of them here. And these processes actually correspond to parameters in the model. For instance, the differentiation of osteoblasts that is affected, well, that is, or the differentiation of chondrocytes, for instance, is, or this is the arrow for the differentiation of of osteoblasts. There is also a problem with the endochondral ossification. That would be this arrow. The fibroblast compartment becomes much more active, which is all the exclamation marks here because they're over activated. So migration is overactivated, proliferation is overactivated. So we can play with these parameter values, or we can simulate these mutations, or the effect of these mutations also in the model. But the problem is we don't really know to what extent these parameters are affected by the congenital disorder. So instead of trying to come up with just one case that was the NF1 healing, which is also not the case in biology because we have a very wide variation in phenotypes, we decided to actually perform sort of exploratory in silico clinical trial where we simulated 200 NF1 patients, where these 8 factors that I mentioned, the parameter values related to those processes were varied between the normal value and either 0 or a maximum value. So to have a scan, if you wish, of the entire diversity that could arise from this NF1 mutation. So we had 200 virtual patients that all had a combination of these 8 parameter values somewhere between the normal situation and the total problematic situation. What you get is, if we pick one where you had a typical NF1 fracture, you see that you get very little bone formation. You do get vascularization, which is also what they see in reality, and you get a large fibrous tissue component. Now we can then look at what the effect is of the different parameter values that we changed. And in order to have a simple measure, a simple output measure, we defined a complexity index, which says that if you have a non-union, if you have a large fibroblast compartment, and you have a lot of fibrous tissue, all these 3 combined, then you will get a high value for complexity index. If you have a non-union, but it seems to be a normal non-union where fibroblast compartment is not overactive, then this will give you a value of complexity index of 0.3, and this will be classified as a normal, a non-NF1 non-union. So if we look at the NF1 non-unions, which are the very high values here, we see that if you look at endochondral ossification, for low values, you actually have always non-unions, but also endochondral ossification, any value of endochondral ossification could lead to a non-union, depending, of course, what the other parameters are doing. Then if you wait a bit longer, if you go from 3 weeks to 5 weeks, you see that some of these dots will be dropping down and will actually, where there is still a non-union here, they will form a union, and so their complexity index will drop. But even if you wait longer, for very low values of endochondral ossification, you never get good healing. The same as for very low values of cartilage formation, you never get good healing. Now, how can we translate that to the patient? Obviously, for the patient, we would like to know we would like to know what is going wrong, but more specifically, how can we help the patient? So what treatment should we offer? And BMP administration is one of these treatments that is often done. So we took these 200 patients and we all gave them a BMP treatment at the start of the healing, at the onset of the healing process. And what you see here is that the complexity index before is the purple with the pink. So you have a large chunk of patients that have a high complexity index, so a non-union, an NF1 non-union. If you give a BMP treatment, you get the blue and purple, which is this part. So you see that there is a shift of a number of patients from a high complexity index to a low complexity index. That's the same as what you can see here in these two graphs. With BMP, you have less patients that have more patients that have a union than without the BMP treatment. Now, if we take a more closer look at these patients and we classify them according to their complexity index without BMP treatment and with BMP treatment, so if we look at the horizontal axis, on the left you have the patients that were healing, and on the right you have the patients that were non-healing. If you look at the vertical axis, at the bottom you have the patients that have healed with the BMP treatment, and at the top you have the patients that have not healed. So if we then look at the quadrants, well, the lower left quadrant, those are the patients that were healing without the BMP treatment, and if you give them BMP treatment anyway, well, they still heal. On the right bottom, you have the responders, because those were the patients that didn't heal before when they did not get BMP treatment, but did heal after receiving a BMP treatment. This is what you want to see for most of your patients. On the top right, you see the non-responders, patients that do not heal without BMP treatment, but also do not heal with BMP treatment. And unfortunately, those patients actually also exist in real life. So if we can mine that information and see what is going on, we can come up with new treatment solutions. But then on the upper left corner, you have the adverse responders. Those are patients that without BMP treatment would have healed, but now when BMP was added at the start of the fracture healing process, actually it pushed the patient towards a non-healing. So that's the adverse responders, and those are also an interesting category because that is what you want to prevent, of course. What we then can do is look at refracture rate. So we have the responders, those are the patients that respond to the treatment. And then we have the non-responders low, those were the patients that were healing anyway. You see that there is a difference in fibrous tissue compartment. The patients that were healing even without treatment, they have a very low or relatively low fibrous tissue compartment, meaning that when there is healing of the bone, it is mostly composed out of bone. So the refracture risk is relatively low. But when you have responders, so patients that would normally not heal but have healed in response to the BMP treatment, you see that they have a lot of fibrous tissue still present, so the refracture risk is quite substantial. And also what you can do is then look at which of the model parameters that we played with is actually a predictor, can be a predictor for the effect, whether they will respond to the treatment or not. And that is something that then can be used to maybe stratify patients prior to treatment. Okay, that concludes the 4 examples, and I don't have very long conclusions, just a few more remarks that I would like to make at the end. So what I've tried to show you is how we develop different tools to actually with different modeling tools to contribute to the whole process of tissue engineering, be it the cells, the carriers, the culture, or the in vivo part. And we want to actually establish in silico as an integral part of the research and development pipeline in tissue engineering. Now, same as with all kinds of in silico medicine applications, this requires an interaction of the different partners and a really integrative approach. And what is also important is that FDA and also now EMA are getting involved. Especially with tissue engineering, the regulatory approval is key and is dictating a lot of the research. And so having FDA saying beforehand, yes, in silico models can be a part of your preclinical dossier is really important for in silico tissue engineering take off as a discipline. And just I wanted to show you this this slide, which is taken from the Medical Device and Innovation Consortium in the States and from the FDA, which shows you that today most of the evidence that you see in preclinical dossiers is coming from laboratory animal and human tests with very small margin for computer. But the prediction of this MDIC consortium is actually that almost half will in the future be coming from the models and computer simulations. FDA has issued guidelines, so if you want to include in silico in your preclinical dossier, they have now explained what they want to see from your model, at least for the medical devices already. And so that is actually the message that I wanted to give, that if we take an integrative approach, we can develop modeling tools that can help in advancing all the different parts of the tissue engineering pipeline. I would like to thank the funding sources for this work and you for your attention.
Thank you for excellent presentation, and I use also the opportunity to wish you a very Merry Christmas and Happy New Year. You actually answered my question. It was about the FDA and European regulations in the field, and perhaps maybe you can also share a little bit more details about, because I know you had an FDA visit recently in Belgium, so maybe also a little bit more details about that. And thank you again.
So Tina Morrison came from the FDA, came to Belgium for the inauguration of the VPHi Alliance, and she presented her view, or the view of the FDA, on computer modeling and simulation. If you go to figshare.com and you type in Tina Morrison, you can find all the presentations that she has given during that visit. So that might be already interesting. What she told us is that for obviously medical devices, they're the furthest advanced, so you have the guidelines already, but they have now established an FDA-wide working group on computer modeling and simulation. And this FDA-wide working group also pertains to pharma and to the ATMP, so the tissue engineering products that we are working on. So they are now, they have established this working group, and they will try to, first of all, see internally to make sure that all the regulators that are dealing with dossiers know enough about modeling so that they can actually assess that properly. But on the other hand, they will also want to issue more guidelines so that this all becomes standardized and streamlined. There is next year, in the spring next year, there will be the VMV40 guidelines that come out. Those are verification and validation guidelines made by the ASME and the FDA together. ASME has these standards for other fields, other engineering fields, but so now for medical devices there will also be the VMV standard, which tells you exactly, or which tells you what to do to make sure that your model is verified and validated. I think that's also something interesting for us to keep an eye on.
Maybe we got something else coming in from Christian. Can you see the question list?
Yes, so the question is, in regards to your model for fracture repair, have you done much in terms of external stimuli? External stimuli, I presume these would be the growth factors and mechanical stimulation. So the growth factors, yes, but very rudimentary, as just BMP, not a type of BMP, just BMP, and not in dosing either. In terms of mechanics, well, that has been very limited, the effort that we did there, because in our tissue engineering applications, we always work with an externally fixated defect. So really the mechanics, the stabilization on the one hand, but then also the stimulation on the other hand, is something that we did not look at much more than that. Well, it was actually Ms. Bava that worked on the ultrasound, and she used our model. So I was also involved in that study. It was mainly Aurélie Carrier that worked together with Maria to redesign, let's say, the model to look also at ultrasound. So it is possible to add these external stimuli, but I'm not working on it personally. So Christian, yeah, and then anybody else, if you want to have more discussions, then we can definitely get in touch. Just drop me an email, and we'll set up a one-on-one meeting. Martina, I don't see any other questions.
Yes, I don't see them either. So, Lisbeth, thank you very much for the great presentation, very detailed. I would like to thank you all for having joined the webinar today. We are planning to have another webinar in a couple of months by James Sharps from Spain. So if you want to receive information about this, you can simply check the VPHi Institute website or subscribe to our monthly newsletter. Great. Thank you, everybody.
Okay.
And thank you, Liz.
Bye, Martina. Bye, guys.
Bye-bye.